cancer impacts a lot of lives, and the ability for scientists and researchers and clinicians to understand exactly what cancer is, and then to develop therapies and treatments for cancer has the potential to impact not only the millions of people who have cancer right now in the United States, but also all of their family and friends. And so cancer is characterized by the uncontrolled growth of abnormal cells. Now, the uncontrolled growth of abnormal cells can lead to the formation of a tumor, and tumors are a mass of tissue that lack normal function, and they cause disease by interfering with the normal function of the neighboring tissue. So we have two major classes of tumors. The first is benign, and so these tumors are localized and lack the capacity to spread throughout the body. So essentially, they cannot metastasize. Our second class of tumors are malignant tumors, and these are ones that have the potential to invade nearby tissues and also spread to other parts of the body. And so when we're talking about cancer, we're actually talking about malignant tumors and not benign tumors. So normal cells grow and divide in an orderly fashion, whereas cancer cells grow and divide uncontrollably. But cancer cells have the potential to invade other tissues and spread throughout our bodies. So cancer is primarily caused by an accumulation of genetic mutations, and these mutations lead to changes in the production or activity level of proteins, and the other is being off or a broken brake pedal. So in our cells, oncogenes are basically genes that are cancer related. So onco is the prefix for cancer or related to tumor. So these are like cancer genes or tumor genes. And in normal cells, these genes help cells grow appropriately when they're turned on at the right times. However, in cancer cells, there are mutations which cause this gene to always be turned on or activated. Tumor suppressor genes are genes which are suppressing tumors. So these are genes which help prevent cell growth or repair DNA damage. In cancer cells, these genes have mutations which cause the gene to be permanently turned off or inactivated. So essentially in cancer cells, oncogenes are always on and signaling for the cell to grow, and tumor suppressor genes are turned off. So the cell has difficulty preventing growth and repairing DNA damage. And so in the average cancer, there are 20 to 80 mutations on average. And one of the things that we're working on is trying to figure out which of these mutations are actually causing cancer. Researchers are using cancer genome sequencing to help understand cancer. And some of our goals are to identify driver mutations as well as weed out passenger mutations. And so what this means is we're looking for the mutations that are really driving or causing cancer. And we're trying to weed those out, weed out the passenger mutations, which are mutations the cells have acquired over time simply because they're abnormal and growing rapidly. And these mutations may not be directly contributing to cancer.
Malignant tumors can metastasize or spread to other parts of the body, and that allows them to cause disease in these new locations as well, which is generally why these are more severe tumors to have. Additionally, this makes treatment more difficult. The potential for metastasis is the definition of malignancy, but luckily for all of us, metastasis is a difficult and rare process, and it's actually possible to have thousands or tens of thousands of cancerous cells circulating in the blood system, and not a single one of them. So our three major current types of treatment are surgery, radiation, and chemotherapy. So surgery is very effective if we have a localized single tumor to work with. Radiation therapy is also helpful for localized treatment, and particularly helpful for cancers that we can't get to surgically. So again, this is a CT scan in the same plane. This is the patient's heart, and the black area is the patient's lungs, and the white mass here is the patient's tumor. The different circles indicate how much radiation we're going to deliver to that tumor. The goal here is to deliver as much radiation as possible to the tumor and as little radiation as possible to the surrounding areas. And the main way that this is done is by delivering radiation from a number of different angles so that it overlaps on the tumor, but the amount through any given surrounding tissue is limited. Now, chemotherapy is a use of drugs to treat cancer, and this is really our major treatment for when cells are in different parts of the body. Now, to think about this, we need to develop drugs which kill cancerous cells but don't affect the normal human cells in the body. The main way that we've done this for decades is to develop drugs that just generally target rapidly dividing cells, because this is the definition of cancer. As you had seen earlier, every time a cell needs to divide, it makes an entirely new copy of its DNA. So we have a variety of drugs which prevent that from happening. And there's also a particular set of mechanisms which need to occur in order for one cell to divide into two. So we also have drugs which inhibit that. Unfortunately, this leads to a number of predictable side effects, which are damages to the other rapidly dividing cells in the body, such as your hair, your skin, the cells lining your stomach and intestines. This is where the nausea and loss of appetite comes from, as well as the cells in the bone marrow. So this is where the fatigue and the susceptibility to infection comes from. So one of our goals going forward, which I'll talk about more, is developing drugs which are more specific for the cancers, which can be helpful in killing the cancers while leaving your normal cells. Fortunately, we're living in a time where we know way more about cancer than we ever did in the past due to new technologies. So the Human Genome Project finished in 2003. We sequenced our first cancer genome in 2006, and by 2015, we've sequenced over 1,000 cancer genomes and parts of many, many more than that. So our knowledge of the changes in DNA which are driving cancer is much, much larger than it has ever been at any point in the past. So where we're headed is an era of personalized medicine. And the idea here is that we treat a patient's tumor based on the specific mutations causing that tumor.